Now we're going to talk about enzymes, what they are and how they work. Okay, so enzymes usually are made of protein. So normally most enzymes are protein. There are some enzymes that are made of RNA, but we don't worry about them right now. We're just looking at enzymes. So there are plenty of reaction that happens in our body, and pretty much all reactions require an initial input of energy, no matter whether it's exergonic or endergonic. Why? Because for a reaction, you're breaking old bonds and you're forming new ones. So breaking bonds is usually something that doesn't happen under conditions that usually living beings live. For example, for us, it's 37 degrees. If it was easy to break the bonds in the chemicals in our body, would be in big trouble. So if you think about it, for example, we have cellulose. When you start a fire, if during fire season we get in trouble because the temperature is so high that and sometimes there is a spark and trees started burning. But usually during normal temperatures, trees don't spontaneously just vanish or catch in fire. So when we want to create firewood, we need a spark, right? We need a spark to start a fire, and that spark provides that initial energy that is required to break old bonds and forming new ones. So that initial energy is called activation energy. So every reaction, no matter whether it's exergonic or endergonic, requires an initial input of energy. Now in this image, you are shown the energy profile for an endergonic pathway, but the same thing is true for exergonic pathways as well. So if you want, you can think about this activation energy as that spark that you need to start the fire. Now, our bodies, as I mentioned, has a lot of chemical reactions in it, but it's not like we can set our bodies on fire and get reactions going. That's just can't be done. Otherwise, we'll just vanish out of existence. So how does nature solve that? Well, enzymes. What enzymes do, they reduce the activation energy that is required for a reaction such that it can happen in, let's say, 37 degrees Celsius or 98.6 degrees Fahrenheit in our bodies. So in here you have two lines for energy profile. The purple one is with no enzyme, and then the red one is with enzyme. So what is the enzyme doing? The enzyme is lowering the activation energy. So you don't need as high a temperature or this big spark and a lighter fluid in order to make this thing happen. Now notice one thing. Enzymes don't change the amount of free energy of the reactants or the products. They only change the activation energy, and that's something that is very, very important to understand. Because what enzymes do, they come in, they work on the reactants, they catalyze the chemical reaction, and then they release the products. Enzymes do not become part of the reactants or part of the products. They just come in, do their thing, and leave. And the only thing that they modify is the amount of activation energy that is required. They lower the activation energy. Enzymes reduce the activation energy of a reaction. Now again, to help you better understand these energy diagrams, I'm going to recreate them for you. So we have this graph again, and let's say we have a reactant here and we have a product that has a higher energy. So the reaction, in order to proceed, has to go through an initial energy input, and then this is the highest energy state that you get, and then it comes down and you reach the product. Sorry if this is looking ugly. It's not supposed to be ugly. So let's fix it. Let's fix it, make it prettier. So we're going to draw this, and it's going to go up here. So this state is called the transition state. That's when old bonds are breaking and new ones are forming. Okay? So now what is the activation energy in this case? The activation energy is from here to here. That's the activation energy that is required. That's the amount of energy is required to make the reaction happen. And what is the difference in the energy level of the reactant and product? This is the reactant, that's the product. This is delta G. Okay? Now, this would be a reaction that does not have an enzyme, but what if we do it in presence of enzyme? I'm going to use another color. So if you remember, the enzyme does not change the activity, the energy level of the reactant. All it's doing is reducing the activation energy. So the energy level of product or reactant doesn't change, but the activation energy now is this low level. Okay? So now let's look at, so what kind of a reaction is this? This is an endergonic. So let's look at another one. We have a reactant and a product. Product has a lower amount of delta G. So then what does it look like? The energy would be, first you have to go through this activation state, and then you go down here. All right. So then what is the activation energy in this case? It's this one. That would be the activation energy. And then the delta G is from reactant to product. That would be delta G. So now, again, how does an enzyme work? An enzyme doesn't change the level of energy of a reactant, just reduces the activation energy. Sorry, let me fix this. Reduces the activation energy, but the amount of free energy associated with reactant and product remains the same. All the enzyme is doing is lowering the activation energy. So then this is minus enzyme. This one green one is plus enzyme. So here's this image is showing you kind of an artist's rendition of what happens during a enzyme catalyzed reaction. Now we talked about enzyme reactions, and we said that there are reactants that are turned into products in chemical reactions. Okay, now enzyme catalyzed reactions specifically work on specific reactants, and that's a good thing. Because if you think about our bodies is full of enzymes, and if all enzymes catalyzed, all reactions worked on all molecules would be in big trouble, because then reactions would go out of control. So enzymes are very specific to particular reactants, which in terms of enzymes, we call them substrates. So each enzyme is specific to a particular substrate in its body, in its shape. It's like it has grooves inside its body that fits the substrate as a lock and key. So the shape of this substrate is nicely fitting within what's called the active site. So the substrate binds to the active site, and you get an enzyme substrate complex. And the enzyme goes under a process called induced fit. Now, induced fit, that means if you compare this enzyme in stage 1 to enzyme in stage 2, you see that this enzyme is kind of a little rounder and it's hugging the substrate. So when the substrate binds to the enzyme, it hugs the substrate here and it changes shape. That's called induced fit. Okay? And then once the substrate forms the enzyme substrate complex, the chemical reaction is catalyzed, and then the enzyme releases the product. Okay? Sometimes enzymes need a little help in order to catalyze reactions, and there's two different kind of entities that help enzymes do that. And they are divided into two factors because one group, which are called cofactors, they're inorganic molecules. Remember, for a molecule to be organic, there has to be both carbon and hydrogen. So molecules like the ion of iron atom and magnesium, these molecules help enzymes catalyze the reactions. And then another class is coenzymes. These are organic molecules. One of the reasons you need to take vitamins is because the enzymes in your body require these coenzymes in order to function. So here are examples of the different dietary vitamins that you take. Vitamin A, folic acid, vitamin B1, vitamin C, B2, D2, B6, and vitamin E. These are all organic molecules, and they help enzymes do their work. Now, our bodies, our cells are very efficient systems. Our bodies and cells have a lot of enzymes in them, but when the function of those enzymes isn't really needed, for example, when an enzyme makes a particular product and when that product is abundant and when there is no need for the cell to make more of it, what the cell does, it reduces or inhibits the activity of that enzyme that produces the product. Conversely, sometimes the cell really needs the activity of an enzyme to be ramped up, or sometimes the cell makes an enzyme and kind of keeps it on call. When somebody's on call, you're really not working, but you're waiting for a phone call to come to say, hey, we need you at work. So similar thing happens sometimes when a cell needs to catalyze a reaction. It makes an enzyme and kind of keeps it in a lower activity state, or in a state that is not quite active, but then when on demand, when that substrate comes in that needs to be broken down, the body increases the activity of the enzyme or activates the enzyme. So we're going to take a look at different types of reducing and activating the enzymes. Let's talk about inhibition of enzyme activity first. There are two classes of enzyme inhibitors, two different ways that scientists have observed that enzymes are inhibited. One is competitive inhibitor and the other one is non-competitive inhibitor. So competitive inhibitor, as the name implies, the competitive inhibitor competes with substrate for binding to the active site. For binding to active site. So therefore, in the presence of a competitive inhibitor, you're going to need more substrate to get the same amount of activity. For non-competitive inhibitor, there is no competition for binding to the active site, and a non-competitive inhibitor binds elsewhere. It binds elsewhere. It does not bind on the active site. And the profile of the way these inhibitors change the activity of an enzyme is a good indication of that. So when you look at the purple line, the purple line is without an inhibitor. When you look at the competitive inhibitor, the maximum enzyme activity, let me change that to green, the maximum enzyme activity doesn't change. So no change in maximum enzyme activity. So it's really this competitive inhibitor is not in any way changing the enzyme in terms of the maximum activity that it can give us. So the only effect is that in the presence of a competitive inhibitor, you're going to need more substrate to get the same level of activity. But when you look at the non-competitive inhibitor, the non-competitive inhibitor actually reduces the maximum enzyme activity. So your maximum enzyme activity is not going to be as high anymore. That's because non-competitive inhibitors actually change the shape of the enzyme. And it substantially changes the way the enzyme functions. So no matter how much substrate you add, you're still not going to get to the same level of activity that you had in the absence of the inhibitor. Now let's try to draw this from scratch so you can maybe understand it better. So this is the graph. This is the substrate concentration. Remember, when we talk about concentration, we put the name of the molecule in brackets, and then on the y-axis is enzyme activity. So in the absence of a substrate, when we keep adding more and more and more substrate, the enzyme activity goes up, 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 up. Finally, at a particular concentration, the enzyme levels off. That's because no matter how much more substrate you add in to the reaction, the enzyme just can't process it fast enough. So that is called Vmax, or maximum activity. And at this point, we say the enzyme is saturated. It just can't work fast enough to process the substrate that you give it. Okay? So now this is, let's say this is the enzyme and that's the substrate. But then imagine that we have a competitive inhibitor that kind of looks the same as the substrate, not quite, but it looks similar such that it can bind to the same site. So if I have an excess of this substrate that can bind to the active site, then the substrate is going to have to compete with it for binding. This is the substrate. Okay? And this is the inhibitor. So then the enzyme is going to have to get more substrate to get the same level of activity. You're going to need more substrate to get the same level of activity. So then the profile is going to be something like this. So at each level of substrate, you're going to need, you're going to get less activity. So if you were to look at this line, for example, just for argument purposes, this would be 10, and let's say this would be 5 level of activity. You see that in the absence of the inhibitor, you need this much substrate, right? But then if you extend this in the presence of the inhibitor, that's the same level of activity, right? But you're going to need more substrate to get the same level of activity. Okay, that's for a competitive inhibitor. For a non-competitive inhibitor, doesn't matter how much substrate you add, you're still not going to get the Vmax because you're fundamentally changing the shape of the enzyme and you're always going to be getting a low level of activity. So then a non-competitive inhibitor would be if this is the enzyme, and then this would be the substrate that's fitting in here. The non-competitive inhibitor isn't going to bind at the active site. It's actually going to bind somewhere else, here, let's say, on the enzyme. And what it's going to do, it's going to change the shape of the enzyme, maybe change the shape of the active site. So the enzyme isn't all as active as it used to be. Now, when an inhibitor doesn't bind the active site, it's said to be an allosteric inhibitor. Okay? And basically allosteric means that whatever molecule is working is not competing with the substrate. So allosteric inhibition or non-competitive inhibition is an inhibitor that is not binding the active site, which is here. So see, it's not binding, it's not competing with the substrate. Now you could also have allosteric activators, and this happens in nature quite a lot. So let's say the original enzyme is made in an inactive state and it's on call. It's on call for the cell to say, okay, I have a situation where you have to now start working. So then in here we have an activator that comes and binds to some place on the enzyme that is not the active site, and it changes the shape of the enzyme such that now it can accept the substrate. In its inactive site, it couldn't accept the substrate. See, the active site wouldn't be able to fit this. So then it activates the enzyme. The activator activates the enzyme by binding to it. The opposite is true for allosteric inhibition. In the absence of the inhibitor, the enzyme was able to nicely fit into with the substrate. But in the presence of the inhibitor, you can see that the shape of the active site has changed, so it really can't bind to the substrate. Feedback inhibition is a very commonly used mechanism for regulating when an enzyme should be active and when it should stop working because it's just producing too much of something. It happens quite a lot in pathways. So what happens in a feedback inhibition, you're dealing with a metabolic process. So this is a pathway. And then this pathway produces an end product. This is an end product that this cell requires. But sometimes this end product is produced in excess. If you have too much of it, then it would be really a waste of resources for the cell to keep making this product. So at some point, as a particular point after, let's say, a tipping point, this end product actually becomes an inhibitor of an enzyme in the pathway. In this particular process, the enzyme comes back and the end product comes back and inhibits the enzyme at the beginning of the pathway. So again, this if you look at it, you will see that this is non-competitive inhibition, which comes in and changes the shape of the enzyme so it cannot process the substrate anymore. And again, this is a very common method of regulating enzyme activity in nature.